Hello everyone, now let's see about Nucleosome, a small illustration video that shows where and how the nucleosome presents. Chromosomes are found inside the nucleus of a cell. What is the structure of a chromosome? A chromosome is a long DNA molecule that coils around some special proteins in a highly organized pattern. The chromosome carries genes, which are segments of the DNA molecule. Genes are the basic unit of heredity. Structure of nucleosome.
A eukaryotic chromosome consists of a linear DNA molecule. This DNA is condensed into a complex structure with histones and non-histone proteins. A nucleosome is a basic structural unit of chromatin, was described by Roger Kornberg in 1974. Nucleosome consisting of the segment of DNA coiled in sequence around 8 histone protein cores. The nucleosome core particle consists of approximately 146 base pairs, BP, of DNA wrapped in 1.67 times left-handed around a histone octamer.
Histone octamer consisting of 2 copies each of the core histones H2A, H2B, H3, and H4. Linker histones such as H1 is bound to the DNA as it enters each nucleosome core particle. Nucleosome core particle along with H1 is known as nucleosome chromatosome. Nucleosome, which consists of 166 base pairs of DNA. Nucleosome 10 nm in size.
Adjacent nucleosomes are joined by a 10 to 80 BP length DNA termed linker DNA. The length of linker DNA, depending on species and tissue type. Histone HL molecules appear to play an important role in this stage of chromatin condensation. Histone H1 protein determining the DNA replication and transcription. Nucleosomes are the fundamental repeating sponheads of all eukaryotic chromatin, except when packaged in sperm. They are made up of DNA and 4 pairs of proteins called histones, and resemble beads on a string of DNA when observed with an electron microscope. They represent the first order of DNA compaction in the chromosome. Formation of nucleosome core particle. It consists of 146 BP of DNA wrapped 1.8 times in a left-handed helix around the outside of an octamer of histones. Chromatosome. The core particle interacts with one molecule of histone H1 to form a particle containing 166 BP of DNA called chromatosome. Nucleosome. The chromatosome links with the linker DNA, forming a nucleosome containing 200 BP of DNA. Chromosomal proteins. The chromosomes of eukaryotes are made up of DNA and proteins. There are 2 major types of proteins associated with DNA in the chromatin. Histone and non-histone. Histone. They are the most abundant proteins associated with the chromosomes. They are very rich in basic proteins. At normal pH of the cell, the histones have net positive charge that facilitates their binding to the negatively charged DNA. This positive charge is found mainly on the amino group of the side chains of the basic amino acids lysine and arginine. Histones lack tryptophan. Each class of histone consists of a N-terminal, which is hydrophobic, C-terminal, which is hydrophilic, central globular structure, which forms the central molecule. H2A, H2B, H3 and H4 together form an octamer, while H1 links to the linker DNA outside the octamer. DNA and core histones are held together by several types of non-covalent bonds, including ionic bonds. The bonds formed between negatively charged phosphates of the DNA backbone and positively charged residues of the histones.
5 major types of histones associated with karyotic DNA are H1, H2, H2B, H3, H4. Functions of histones depress the genetic activity. As the histones increase the compaction of the DNA, it depresses the genetic activity. Structural role. They play a structural role in the packaging of DNA molecules and hence render them more compact. Non-histone. They are all the proteins associated with the DNA apart from the histones. They are very different from histones. They are acidic proteins, that is, have a net negative charge and like to bind the positively charged histones. Functions of non-histones. Structural. They play a structural role in the shape of the chromosome. Regulatory. They positively regulate the gene expression and stimulate genetic activity. They play an essential role in the transcriptional and translational activity. Enzymatic. Many enzymatic activities are associated with chromatin. Enzymes at chromosomal metabolism, nucleic acid polymerases, nucleases and enzymes at histone metabolism are all non-histone proteins. Interaction between the DNA and histones. Interaction between the DNA and histones take place between negatively charged phosphates of DNA and positively charged groups of histone. The major electrostatic interaction of DNA phosphates are with the globular part of the core. The other interactions include hydrogen bonding between oxygen phosphates of the DNA and histones. Thus this shows that DNA doesn't appear to be buried into the core, but contacts it at widely separated points. Thank you. See you in next video.